This is the big one. Do we even start reading the gene? Yes or no? Then post-transcriptional control. This is like the editing room for the RNA message.
Yeah.
How do we process it, stabilize it, maybe even silence it?
Okay. Editing.
Got it. Third, translational control. Now we're at the assembly line. How fast do we actually build the protein from that message? How many copies? Fourth is post-translational modification. The protein's built, but it's often not ready. It needs, finishing touches, activation, targeting, maybe even a tag for destruction.
Activation, tagging. Okay. And the last one.
And finally, number 5 is feedback regulation. How does the whole system adjust itself based on what's happening? How does it all network together?
That really brings us back to that opening analogy, the symphony one. It's a good one to keep in mind.
It really is.
Every single cell, basically, has the same DNA. Billions of base pairs. But a neuron acts totally differently than a muscle cell.
Right. And the difference isn't the musical notes, that's the DNA. It's which instruments are playing, when they play, how loud.
Gene expression is the conductor.
Precisely. It's orchestrating that whole complex tissue-specific performance.
Mm-hmm.
So let's start by actually looking at the sheet music itself. What does a typical eukaryotic gene look like?
Right. Before the conductor can do anything, you need the score. And in our cells, eukaryotic cells, the genes aren't just simple, continuous blocks of code. They're broken up.
They are, interrupted by these long stretches that don't actually code for protein. And understanding this structure, this anatomy, is absolutely key to understanding the regulation.
So where do we start on this map? What are the key landmarks?
Okay, the most crucial starting point is the promoter. Think of it as the big flashing start transcription here sign for RNA polymerase, the copying enzyme. It sits just upstream, just before the gene actually starts. And very often it includes that famous sequence, the TATA box, usually about maybe 25 base pairs upstream. That's a critical landing pad for the basic machinery.
But it's not just about what's right next to the start site, is it? There are these other regions, sometimes really far away.
Exactly. That's where enhancers and silencers come in. These can be thousands of base pairs away, sometimes even tucked inside another gene entirely, but they have a huge impact.
How does that even work if they're so far away?
It's all about the 3D structure of DNA in the nucleus. DNA is flexible, like a piece of string. Enhancers are sequences that bind specific proteins, and they dramatically boost transcription. They turn the volume way up. They do this because the DNA actually forms a loop, bending over so that the enhancer region with its bound proteins physically touches the promoter area.
Wow, okay. So the DNA literally ties itself in the knot, almost, just to connect those regions.
Pretty much. It's a complex architectural feat. And then silencers do the opposite. They bind proteins that turn the volume down, often by helping to pack the DNA more tightly in that area, making it harder to access. This looping allows for incredibly specific control in different cell types.
Okay, looping for long-distance control. Now, what about the parts within the gene sequence itself? The famous exons and introns. This confuses a lot of people.
Right, so exons are the sequences that actually exit the nucleus and get translated into protein. They're the important bits, the paragraphs that make it into the final essay, so to speak.
And introns.
Introns are the intervening sequences. They get transcribed into the initial RNA, but they have to be cut out before the message goes to the ribosome. They're like the rough draft sections you delete. And the fact that we have introns and need to remove them opens up a huge regulatory possibility we'll get to later, alternative splicing.
Okay, exons are kept, introns are out. But there are also bits at the very beginning and end of the RNA message that aren't protein code either, the UTRs.
Yes, the untranslated regions, or UTRs. There's a 5-year UTR at the beginning and a 3 UTR at the end. Think of them like the margins and footnotes of the message. They don't code for amino acids, but they are packed with regulatory information. The 5-year UTR controls how easily the ribosome grabs on and starts translating. The 3-year UTR is crucial for determining how long the mRNA lasts its stability, and it's also where tiny regulatory RNAs, like miRNAs, often bind.
So the 3-yuck UTR is like a timer, almost.
In a way, yes. Specific sequences there can mark the mRNA for quick destruction. It's really important for controlling how much protein gets made from a single message.
And finally, how does the copying machine know when to stop, the end signal?
That's the poly-A signal. It's typically the sequence ATAS in the DNA. It tells the transcription machinery, okay, stop copying soon, and it also signals where the protective poly-A tail needs to be added later on to the RNA. It's like the period at the end of the sentence.
Okay, so we've got the gene structure mapped out. Now we're at control point 1, transcriptional control. This is arguably the most fundamental step. If you don't start transcription, nothing else follows.
Exactly. This is the main gate. And the gatekeepers here are proteins called transcription factors, or TFs.
These are the proteins that actually stick to the DNA.
Yes, they bind to specific DNA sequences, usually in or near the promoter or those enhancer silencer regions, and their job is either to help RNA polymerase get started or to block it. They're the primary decision makers.
And it's important to know there isn't just one kind of TF, right? There are different levels.
Precisely. You've got the general TFs. These are things like TFII, TFIB, TFIIB, a whole crew. Think of them as the baseline machinery. They're needed for pretty much all protein-coding genes to get transcribed at a basic level. They assemble right at the promoter, often around the title box.
Okay, the basic crew. But then there are others.
Then you have the specific TFs. There are thousands of these. These are the specialists. They get activated by specific signals, maybe a hormone, maybe a growth factor, and they bind to unique DNA sequences, sometimes far away at enhancers. They're the ones that turn specific genes on or off really strongly, driving cell differentiation and responses.
What makes them so effective? I remember reading they have sort of distinct parts, like tools for different jobs.
Oh, that's a great way to put it. They generally have at least two key domains. One is the DNA binding domain, which is shaped very precisely to recognize and latch onto a specific DNA sequence. We see characteristic structures here, like the helix, turn helix motif, which is really common in developmental genes, or the zinc finger, which uses a zinc ion to stabilize its shape for DNA binding. You see those in hormone receptors a lot. And the leucine zipper, which allows two TF proteins to kind of zip together, forming a dimer that binds DNA much more strongly or specifically.
So one part sticks to the DNA. What does the other part do?
The other key part is usually the activation domain, or sometimes a repression domain. This is the part that interacts with the general transcription factors or other regulatory proteins, basically telling the RNA polymerase machinery to go or stop.
Okay, but how does the cell control these specific TFs? They can't just be active all the time, right? Otherwise, a liver cell might start trying to be a neuron.
Absolutely not. Their activity is tightly controlled, usually through signal transduction pathways. It starts with a signal from outside the cell, or maybe inside. This signal triggers a cascade. Often this involved activating enzymes called protein kinases, things like pKa or the MAPK pathway components. These kinases add a phosphate group to the transcription factor. It's called phosphorylation.
Ah, the phosphate switch again.
It's everywhere. That phosphate group acts like a switch. It changes the TF's shape, maybe allows it to enter the nucleus if it wasn't there already, or enables it to bind DNA effectively. Suddenly it's active and can turn its target genes on, and importantly, it's reversible. Phosphatesis can remove the phosphate to turn it back off. Hashtag B epigenetic regulation, the DNA memory.
So TFs are like the fast-acting switches, often responding to immediate signals. But there's a deeper level of control, right? Something more about the fundamental accessibility of the DNA itself, epigenetics.
Exactly. Epigenetics is fascinating. It's about heritable changes in gene function that don't involve changes to the actual DNA sequence. It's like adding sticky notes or highlighting to the DNA blueprint that tells the cell whether to read certain sections or not. It's a form of DNA memory.
And a lot of this comes down to how the DNA is packaged with those histone proteins.
Precisely. Our DNA isn't just floating around. It's wrapped around these spool-like proteins called histones, forming structures called nucleosomes. Think of it like thread on a spool. If that thread is wound super tightly, the transcription machinery physically can't get access to the genes in that region. The gene is effectively OFF. Epigenetic marks control how tightly that thread is wound.
Okay, so what's the main tighten the thread signal, the OFF signal?
The classic silencing mark is DNA methylation. This is where enzymes called DNMTs, DNA methyltransferases, add a small chemical tag, a methyl group, directly onto cytosine bases in the DNA, usually where a C is followed by a G. These are called CPG islands. When you see a promoter region that's heavily methylated, that gene is almost always silenced, shut down, OFF. Think M for methylation, M for mute. It's often a very stable, long-term silencing mechanism.
Okay, methylation means OFF. What's the corresponding on-end signal? How do you loosen the thread?
The primary activating mark is histone acetylation. Here, enzymes called HATs, histone acetyltransferases, add an acetyl group to the histone proteins themselves, specifically to lysine amino acids in their tails. These histone tails normally have a positive charge, which helps them cling tightly to the negatively charged DNA. Adding an acetyl group neutralizes that positive charge.
Ah, so it weakens the grip.
Exactly. It loosens the electrostatic attraction. The DNA unwinds a bit from the histone spool, making it more accessible to transcription factors and RNA polymerase. So think A for acetylation, A for active, gene on-end.
Which means the enzymes that remove those acetyl groups, the HDACs, histone deacetylases, must be responsible for tightening it back up and turning genes on off again.
You got it. HDATs acetylate ON, HDACs deacetylate ONFF. This constant back and forth is a major way the cell controls the local chromatin structure. We call the tightly packed, silenced chromatin heterochromatin. It looks dense under a microscope. The loosely packed, accessible active chromatin is called euchromatin.
So it's chemical tags. But is there also, like, physical pushing and shoving involved, actually moving those histone spools around?
Oh, absolutely. There are large protein complexes called chromatin remodeling complexes. A famous example is Swesio-DNF, pronounced switch sniff. These are like molecular bulldozers. They use energy from ATP to physically slide nucleosomes along the DNA or even completely eject them from a region. This is crucial for opening up a specific gene promoter very quickly when it needs to be accessed.
This whole system of packaging and marking,
Hmm.
it sounds like it could easily go wrong in diseases like cancer.
It's fundamental. In many cancers, genes that should be stopping tumor suppressor genes get silenced because their promoters become abnormally hypermethylated. Too many OF signals. Conversely, genes that drive cancer growth oncogenes might become hypomethylated, losing their LVEF signals and becoming overactive.
And this actually leads to treatments.
Yes. For example, HDAC inhibitors are a class of cancer drugs. They block the HDAC enzymes, preventing them from removing those activating acetyl marks. The hope is to force cancer cells to re-express silenced tumor suppressor genes, putting the brakes back on growth. #TAG-C hormone response elements.
We've talked about signals from outside activating kinases and epigenetic marks at setting the baseline, but what about signals that just walk right into the cell, like steroid hormones? How do they control transcription?
Oh, yes. Steroid hormones like estrogen, cortisol, testosterone, thyroid hormone. These guys are different. They're small and lipid-soluble.
So they don't need a receptor on the cell surface?
Nope. They just diffuse straight through the cell membrane. Easy access. Once inside, they find their specific intracellular receptor, which might be floating in the cytoplasm or already in the nucleus.
And what happens when the hormone binds the receptor?
The binding causes the receptor protein to change shape. And here's the key. The activated hormone receptor complex is itself a specific transcription factor, then travels directly to the DNA and binds to specific sequences called hormone response elements, HREs, located near the genes it controls. This binding directly turns those target genes on, or sometimes OFF. It's a very direct pathway for widespread, often long-lasting changes in gene expression, coordinating things like metabolic shifts or developmental processes.
Huh.
Okay, so transcription has started. The RNA polymerase has made a copy of the gene. We have what's called a pre-mRNA transcript, but it's definitely not ready to leave the nucleus yet.
Right, it needs work. This is control point 2, the editing room. What happens here?
Several crucial things. First, the pre-mRNA needs some protection and identification tags.
It's like a cap and a tail.
Exactly. At the very beginning, the 5-arit, the cell adds a special 5-arit cap. It's a chemically modified guanine nucleotide called 7-methylguanosine, or MG. This cap does two vital jobs. One, it protects the RNA from being chewed up immediately by enzymes called exonucleases. Two, it's the signal that the ribosome recognizes later on to know where to start translation. No cap, no protein.
Okay, protective cap at the front. What about the back end, the tail?
At the 3-arit, an enzyme adds a long string of adenine bases, maybe 50 to 250 of them. This is the famous poly-A tail. The poly tail also helps protect the mRNA from degradation, and generally, the longer the tail, the longer the mRNA lasts in the cytoplasm, meaning more protein can be made from it. It's also important for getting the mRNA exported out of the nucleus.
Cap on the front, tail on the back. But the really complicated part is getting rid of those introns, right? The splicing.
Oh yeah. Splicing is an incredibly precise molecular surgery. Remember, the exons are the parts we want to keep, and the introns are the intervening parts we need to remove. This is carried out by a huge dynamic machine called the spliceosome. It's made up of several small RNA molecules complex with proteins. These are called SNRNPs, small ribonucleoproteins, or people often call them SNRPs.
SNRPs. Okay, how do they know where to cut?
The spliceosome recognizes specific short sequences at the boundaries between exons and introns. There's a 5-ORI splice site, usually with a GU sequence, a 3-ORI splice site, usually AG, and an important internal site called the branch point, which contains a specific adenine nucleotide.
So it finds those 3 signals.
Right. And then, through a series of RNA-catalyzed reactions, it cuts the intron out, forming a loop structure called a lariat. Then it precisely stitches the 2 flanking exons together. If it's off by even one single base, the whole protein reading frame gets messed up downstream. It has to be perfect. #B alternative splicing, and protein diversity.
Okay, wait, let's pause on splicing. Because this isn't just about removing junk, is it? This is where the genome gets incredibly clever. How can we have only about 20,000 genes but make, like, over 100,000 different proteins?
This is it, alternative splicing. It's probably one of the most important ways eukaryotes generate complexity. It means the spliceosome doesn't always cut and paste the same way. In different cell types or under different conditions, the spliceosome can be guided to skip certain exons or use alternative splice sites.
So it can treat an exon like an intron sometimes, just cut it out, along with the actual introns.
Exactly. So from one single pre-mRNA transcript copied from one gene, the cell can generate multiple different mature mRNAs by including or excluding different combinations of exons. Each of these different mRNAs will then be translated into a distinct protein version called an isoform. These isoforms might have slightly different functions or go to different locations in the cell. It's a massive source of protein diversity.
Can you give a concrete example?
The classic one is the tropomyosin gene. Tropomyosin is a protein involved in muscle contraction and cell structure. From this single gene, through alternative splicing, our bodies make over 40 different tropomyosin isoforms. There are specific versions for smooth muscle, skeletal muscle, fibroblasts, brain cells, all tailored for the specific job needed in that tissue, all from one gene.
That's incredible efficiency. Okay, besides splicing, is there any other editing of the RNA message itself?
Yes, there's also RNA editing. This is less common than splicing, but still important. It involves enzymes actually changing specific bases in the mRNA sequence after it's been transcribed. The most common type in humans is I editing, where an enzyme called ADAR converts specific adenosine bases into inosine.
Why inosine?
Because the translation machinery reads inosine as if it were guanosine. G. So changing an A to an I can actually change the codon, potentially swapping one amino acid for another in the final protein. This happens a lot in the brain, particularly with receptors for neurotransmitters, subtly tweaking their function. #CmRNA stability and RNA interference, RNAi. All right, so our mRNA is capped, tailed, spliced, maybe edited. It gets exported to the cytoplasm. But its journey isn't over. How long it survives out there is another control point.
Right, because the longer it hangs around, the more protein gets made from it. So what determines its lifespan?
We mentioned the poly-A tail generally, longer tail, longer life. But there are also specific sequences, usually in the 3R UTR, that act as instability signals. Very common are AU-rich elements, AREs. mRNAs that code for proteins that need to be made only transiently, like inflammatory signals, cytokines, or proteins that drive cell growth, often have AREs in their 3' UTR. These sequences attract specific proteins that trigger rapid degradation of the mRNA. Quick on, quick off.
Okay, so built-in self-destruct sequences. But what about more targeted silencing? Shutting down one specific mRNA. That's RNAi. Right?
Exactly. RNA interference, RNAi, is a really elegant mechanism for sequence-specific gene silencing, using small RNA molecules as guides. The main players we think about in humans are microRNAs, miRNAs. These are short RNAs, about 22 nucleotides long, that are actually encoded by our own genome. They typically target sequences in the 3' UTR of specific mRNAs.
How does the whole process work? There are enzymes involved, like Dicer and RISC.
Right, so miRNAs start out as longer precursors. An enzyme called Dicer acts like molecular scissors, chopping the precursor into the short, mature, double-stranded miRNA. Then one strand of this miRNA, the guide strand, gets loaded into a large protein complex called RISC, which stands for RNA-induced silencing complex.
So RISC is like a patrol unit with the miRNA as its target description.
That's a perfect analogy. The RISC miRNA complex then floats around the cytoplasm, scanning mRNAs. If it finds an mRNA with a sequence complementary to its guide, miRNA, usually in the 3' UTR, it binds. Depending on how perfect the match is, this binding can either physically block the ribosome from translating the mRNA, or it can trigger enzymes within RISC to actually cut the target mRNA in half, leading to its destruction. Either way, protein production from that target gene is shut down.
And this isn't just some obscure mechanism, it's clinically relevant.
Hugely relevant. Dysregulation of miRNAs is involved in many diseases, especially cancer. For example, a specific miRNA called miR21 is often found at high levels in tumors, where it silences multiple tumor suppressor genes. Conversely, we can now design artificial small RNAs to target disease-causing genes. The FDA approval of patisiran in 2018, the first RNAi-based therapy, was a landmark moment. It uses RNAi to silence a gene responsible for a rare inherited nerve disease. The whole new way to make drugs.
Okay, the mRNA has run the gauntlet. It's processed, protected, it's out in the cytoplasm, the ribosome is ready to go. But even now, the cell can still say, hold on, this is control point 3, translational control. controlling the actual protein synthesis rate.
That's right. Just because the mRNA is there doesn't mean it automatically gets translated at full speed. The cell can regulate how efficiently ribosomes initiate translation and how fast they move along the message. Hashtag A, initiation control.
And where's the main choke point for controlling translation?
The major control point is usually initiation getting the ribosome assembled correctly on the mRNA and finding the start codon. This is often the slowest, the rate-limiting step. And the cell has a powerful way to slam the brakes on globally if needed. The key player here is a protein called EIF2, eukaryotic initiation factor 2. Its job is to bring the very first tRNA, carrying methionine, to the small ribosomal subunit.
Okay, EIF2 is crucial for starting. How does the cell shut it down?
Usually under stress, right? Exactly. When the cell experiences stress like starvation, viral infection, or if lots of misfolded proteins are accumulating in the ER, it activates specific protein kinases. These kinases phosphorylate EIF2. Once EIF2 has that phosphate group attached, it gets stuck in an inactive cycle. It can't be recycled to bring another initiator tRNA to the ribosome. The result, a rapid and dramatic shutdown of most protein synthesis throughout the entire cell.
Oh, why do that? Why shut everything down?
It's a crucial survival mechanism. Making proteins consumes a huge amount of energy and resources. Under severe stress, the cell needs to conserve energy and focus on immediate survival tasks, like repairing damage or fighting off a virus, not building routine proteins. Hashtag B, regulatory elements in the mRNA.
Okay, that's the global emergency brake. But cells also need finer control over specific proteins, like managing iron levels you don't want too much or too little. How does that work at the translation level?
The iron example is perfect. This involves specific structures within the mRNA itself called iron response elements, IREs. These are little stem loop shapes found in the UTRs of mRNAs for proteins involved in iron metabolism, like ferritin, which stores iron, and the transferrin receptor, which imports iron.
So let's take ferritin, the storage protein. What happens when iron is low?
When iron levels are low, a specific protein called the IRE binding protein, IREBP, is active. It binds directly to the IRE located in the 5' UTR of the ferritin mRNA. This binding physically blocks the ribosome from assembling or scanning properly. Result, ferritin translation is O off F. Makes sense you don't need to make the storage protein if there's no iron to store.
Okay, block is on when iron is low. What if iron levels rise?
When iron levels are HEH, the iron atoms themselves bind directly to the IREBP. This causes the IREBP to change shape and fall off the ferritin mRNA. The block is removed. The ribosome can now access the start codon, and ferritin protein is synthesized rapidly to safely store the excess iron. It's an incredibly elegant feedback loop controlled directly by the molecule iron being regulated.
That is really clever. Protein binding based on iron levels controls the faucet. Are there other ways mRNAs control their own translation?
Yes, another interesting mechanism involves URFs, which stands for upstream open reading frames. These are short little coding sequences found in the 5' UTR before the main protein coding sequence starts. Sometimes a ribosome will start translating one of these URFs. Depending on the specific URF and cellular conditions, this can cause the ribosome to stall, or maybe even fall off the mRNA before it ever reaches the real start codon for the main protein.
So it's like a decoy start
Uh,
or a quality control check.
Sort of. It acts as a way to limit or fine-tune the amount of the main protein that gets produced. It adds another layer of regulation right at the translational level.
And we should probably mention, just for completeness for you, the learner, that bacteria have their own unique trick here, right? Something without proteins.
Oh, absolutely. We have to mention bacterial riboswitches. These are amazing. Parts of the bacterial mRNA itself, usually in the 5' UTR, can directly bind small molecules like a vitamin or an amino acid. This binding causes the mRNA structure to physically change its fold. This change can either hide or reveal the ribosome binding site, the Shine-Dalgarno sequence in bacteria. So the mRNA itself acts as the sensor and the switch, controlling its own translation without needing any regulatory proteins. It's incredibly efficient.
All right, we've made it through transcription, processing, and translation control. The ribosome has done its job, and we have a polypeptide chain of freshly made protein, but we're still not done. Control point 4, post-translational modifications, or PTMs.
Exactly. Just coming off the ribosome, most proteins are, well, inert. They need modifications to become active to be sent to the right place or sometimes to be marked for destruction. It's like assembling a product but still needing to add the batteries or the shipping label.
There must be hundreds of different PTMs.
Oh, hundreds. It's an incredibly diverse layer of regulation. But let's focus on the major ones. The absolute king of PTMs, is phosphorylation.
We keep coming back to this one, adding a phosphate group.
Yes. Done by kinases, removed by phosphatases. Adding that bulky, negatively charged phosphate group drastically changes a protein's shape and therefore its activity. It's the cell's main way of turning proteins on or off rapidly in response to signals. Think of all those signaling cascades, MAPK, PKA, they run on phosphorylation.
Okay, phosphorylation is the main on-off switch. What about marking a protein for removal?
That's ubiquitination. This is where the cell attaches a small protein called ubiquitin, or sometimes a chain of ubiquitins, onto the target protein. That ubiquitin tag is essentially a molecular kick-me sign. It signals that this protein is destined for destruction by the cell's garbage disposal, the proteasome.
How does the cell know which proteins to tag with ubiquitin?
That specificity comes from a large family of enzymes called E3 ubiquitin ligases. Each E3 ligase recognizes specific target proteins, often only when those targets are themselves modified, like phosphorylated or misfolded. This precise targeting is crucial for controlling levels of key regulatory proteins, especially those involved in the cell cycle. Get it wrong, and you can get cancer.
So mistakes by those E3 ligases or mutations in them could be really dangerous, either destroying something vital or failing to destroy something harmful.
Absolutely. If an E3 ligase wrongly targets a protein you need, you lose its function. If it fails to target a protein that should be destroyed, say, a protein-pushing cell division, that protein accumulates and can drive disease.
Yeah.
Finding drugs that modulate specific E3 ligases is a huge area of research right now.
Okay, phosphorylation for activity, ubiquitination for destruction. What about other key modifications, things needed for structure or location?
Well, glycosylation is a big one. This is the addition of complex sugar chains, usually happening in the ER and Golgi. It's essential for the proper folding of many proteins, especially those destined for the cell surface or secretion. It also plays key roles in cell recognition. The ABO blood groups are determined by different glycosylation patterns on red blood cells. And then there's proteolytic cleavage. Sometimes proteins are made as longer, inactive precursors called proproteins or zymogens. Specific enzymes called proteases then cut them at precise points to release the shorter active form. Think of activating digestive enzymes like trypsinogen into trypsin, or processing proinsulin into mature insulin. Hashtag SHAB-B, misfolding and targeting.
So PTMs help activate and direct, but underlying all of this is the assumption that the protein folds into the right 3D shape in the first place. What happens if it doesn't?
That's a major problem. Protein folding is incredibly complex, and if it goes wrong, the protein might not function, or worse, it might clump together into toxic aggregates. The cell has quality control machinery, including chaperone proteins like HSP70 and HSP90, that help newly made proteins fold correctly and prevent them from clumping. But sometimes this system gets overwhelmed or fails.
And that failure leads directly to disease.
Yes, many devastating diseases are linked to protein misfolding and aggregation. Think of Alzheimer's disease, with its plaques of misfolded amyloid beta and tangles of tau protein, or Parkinson's disease, characterized by aggregates of alpha-synuclein. And a really important example connecting back to PTMs and processing is cystic fibrosis. The most common mutation causes a slight misfolding of the CFTR protein. The protein could still function, but the cell's quality control system in the ER recognizes it as defective and tags it for ubiquitination and destruction by the proteasome before it ever reaches the cell membrane. The disease isn't caused by a lack of the protein being made, but by it being destroyed prematurely due to a folding trafficking defect.
Wow, so the quality control is actually causing the problem in that case. Okay, assuming a protein does fold correctly, how does it get to the right place, the nucleus mitochondria outside the cell?
That's protein targeting, and it relies on specific address labels or signal sequences embedded within the protein's amino acid sequence. For instance, proteins destined for secretion or insertion into membranes usually have an N-terminal signal peptide that directs the ribosome to the ER. Proteins going to the mitochondria have a mitochondrial targeting sequence. Those headed for the nucleus need a nuclear localization signal, NLS, usually a stretch rich in basic amino acids. Others have signals for peroxisomes, etc. Each compartment has its own zip code, #C, protein degradation systems. We've touched on protein destruction via ubiquitination and the proteasome. Let's just clarify the main systems the cell uses to clear out unwanted or damaged proteins.
Right. We have the proteasome for individual tagged proteins?
Yes, the proteasome is highly selective. It's like a sophisticated shredder for specific documents marked with that ubiquitin tag. It unfolds the protein and chops it into small peptides, crucial for regulating the levels of specific signaling proteins and cell cycle regulators.
But what about bigger things, like a whole damaged mitochondrion or large clumps of aggregated protein? The proteasome can't handle that, can it?
No, for bulk clearance or large structures, the cell uses a different process called autophagy, which literally means self-eating. In autophagy, a double membrane grows and engulfs the target, whether it's a worn-out organelle or protein aggregates forming a structure called an autophagosome. This autophagosome then fuses with a lysosome, which is basically the cell's recycling center, filled with powerful digestive enzymes. Everything inside gets broken down.
So autophagy is more like taking out the whole trash bin, not just shredding one paper.
Exactly. It's essential for clearing damaged components, recycling nutrients during starvation, and even fighting off some intracellular pathogens.
And knowing about these systems gives us clinical tools.
Definitely. We mentioned HDAC inhibitors targeting epigenetics. For degradation, proteasome inhibitors like the drug bortezomib are used very successfully to treat multiple myeloma, a type of blood cancer. These cancer cells rely heavily on the proteasome to get rid of proteins that would normally stop their growth. Blocking the proteasome causes these regulatory proteins to build up, essentially forcing a cancer cell to choke on its own waste and undergo apoptosis. Targeting autophagy is also being explored for various diseases.
Okay, we've gone through the eukaryotic checkpoints in detail. Now, for you, the learner, it's really important to contrast this with the simpler, faster system used by bacteria. We need to talk about the operon model.
Absolutely. Comparing prokaryotic and eukaryotic regulation really highlights why eukaryotes need all that complexity. Bacteria live in rapidly changing environments. They need to respond fast. The operon is their solution for coordinating gene expression. An operon is a cluster of genes that all work together in a single pathway, like digesting a specific sugar, and they're all transcribed together from one promoter into a single mRNA molecule.
So one message contains the instructions for multiple proteins.
Yes, that's called a polycistronic mRNA. It's super efficient. Let's look at the two classic examples everyone learns. First, the lac operon.
The lac operon, that's for digesting lactose, milk sugar, and it's inducible, meaning it's usually OFF but can be turned on.
Correct. The goal is only make the lactose-digesting enzymes when lactose is actually present and when glucose, the preferred sugar, is absent. It uses a combination of negative and positive control.
Okay, break that down. Negative control.
Negative control comes from the lac repressor protein. It's encoded by a separate gene, LAC-I. Normally, this repressor binds to a specific DNA sequence called the operator, which overlaps with the promoter. This physically blocks RNA polymerase from transcribing the operon genes, LAC-Z, LAC-C, LAC-A.
So the default is OFF because the repressor is blocking the road. How does lactose turn it on?
When lactose is present, some of it gets converted into a slightly different molecule called allolactose. Allolactose acts as the inducer. It binds to the repressor protein, causing the repressor to change shape and fall off the operator DNA. The roadblock is removed.
Okay, block removed, but you said there's positive control too, linked to glucose.
Right. Bacteria prefer glucose. If glucose is available, they don't want to waste energy on lactose, even if it's there. This is controlled by HEAP, catabolite activated protein, and a small molecule called CANMP. When glucose levels are LOW, KMP levels inside the cell go HIGH. CANMP binds to TCAC, and the KCAP-UCAMP complex then binds to a site near the lactor motor. This binding actively helps RNA polymerase bind more strongly and transcribe the operon much more efficiently. It's an accelerator pedal.
So you need the break-off, repressor removed by lactose, and the accelerator press, CMP down because glucose is low, to get maximum speed.
That's exactly. Let's run through the scenarios quickly. 1. No lactose, no glucose. Repressor is down, brake on end. CMP is high. CMP binds the accelerator, but doesn't matter because the brake is on. Result, almost NO transcription.
That makes sense.
2. Lactose present, no glucose. Repressor falls off, brake arrest.
Yeah.
CMP is high. TMP binds, accelerator in. Result, high GH level of transcription. The cell goes full throttle on lactose digestion.
Ideal conditions.
3. Lactose present, glucose present. Repressor falls off, break off value. But glucose is present, so KMP is LW. CMP cannot bind effectively. Accelerator OF. Result, low level of transcription. The cell uses glucose first.
Glucose preference wins.
4. No lactose, glucose present. Repressor is BMD, break on. CMP is low, accelerator off anyway. Result, almost NO transcription.
Okay, that logic is clear. Negative and positive control working together. What's the other classic operon, the opposite type?
That's the TRAP operon for tryptophan synthesis. This one is repressible. The goal is to make tryptophan, an amino acid, but only when the cell doesn't have enough of it already.
So the default state is on-in.
Yes. The genes for making tryptophan are usually being transcribed. The repressor protein for the trip operon is made in an inactive form. It can't bind the operator by itself.
How does it get turned off FF then, when there's plenty of tryptophan?
Exactly. When tryptophan levels get HIGH, tryptophan itself acts as a co-repressor. It binds to the inactive repressor protein. This binding activates the repressor, allowing the repressor tryptophan complex to bind to the triple operator. Once bound, it blocks RNA polymerase, shutting down transcription of the tryptophan synthesis genes. It's classic feedback inhibition. The end product of the pathway shuts down its own production. #B prokaryotic versus eukaryotic comparison.
Okay, understanding both lac and tripoperons is key. Now let's explicitly compare the bacterial world with the eukaryotic world we spent most of our time on. This comparison is crucial for synthesizing the information, especially for you studying this material.
It really is. The differences stem from fundamentally different needs and structures. DNA structure: bacteria have a single circular chromosome, mostly naked DNA, floating in the cytoplasm. Eukaryotes have multiple linear chromosomes, tightly packaged with histones, inside a nucleus.
That location difference is huge too.
Location: in bacteria, transcription and translation happen simultaneously in the cytoplasm. They're coupled. Ribosomes jump onto the mRNA while it's still being made. In eukaryotes, transcription happens in the nucleus, mRNA processing happens in the nucleus, then the mature mRNA is exported to the cytoplasm for translation. They're separated in space and time.
And the way genes are organized.
Gene organization: bacteria use operons, leading to those polycistronic mRNAs. Eukaryotes generally have individual genes, each with its own promoter, producing monocistronic mRNAs. Plus, eukaryotic pre-mRNA needs extensive processing, capping, splicing, polyadenylation, which bacteria don't do.
So the complexity of regulation must be vastly different.
Immensely. Regulation complexity: bacteria rely mostly on transcriptional control via operons. It's fast and efficient for responding to environmental changes. Response time is often in minutes. Eukaryotes regulate at multiple levels: epigenetic, chromatin; transcriptional, TFs, enhancers; post-transcriptional, splicing, RNAi stability; translational initiation, URRFs; and post-translational, PTMs, degradation. This allows for much finer control needed for development and maintaining different cell types, but the response time is much slower, often hours or even days for big changes.
And this all ties back to how much of the genome actually codes for stuff, gene density.
Exactly, gene density. Bacterial genomes are packed, maybe 90% codes for protein or RNA. Eukaryotic genomes